Good evening to all of you, and thank you for joining us to this new webinar of the COVID-19 series organized by the Argentinian Society of Immunology. Mariana Macioni, the Vice President, and Carolina Mezpa, who will chair the webinar, want to remind you that you can make your questions through the YouTube channel. If you prefer, you can make your questions in Spanish, and we will translate them. We have a great pleasure today, and we are very excited to learn about her research because we will count today with Dr. Akiko Iwasaki. Dr. Iwasaki was born and raised in Japan, and then she moved to Canada, where she received her PhD from the University of Toronto in 1998. She made her postdoctoral training at the National Institute of Health in the USA, and then she moved to Yale University as a faculty in 2000. She is currently a professor of immunobiology and molecular, cellular, and developmental biology and a Howard Hughes Medical Institute investigator. Dr. Iwasaki has made seminal contributions to the understanding of how innate recognition of viral infections leads to the adaptive immune response and how adaptive immunity mediates protection against subsequent viral challenges. Her discoveries have led to the way to understand the immune response and to important pathogens with major implications for basic science and medicine. Her fundamental contributions are reflected in the honors and awards she received and by a number of highly cited publications in top journals. Dr. Iwasaki was elected to the National Academy of Science in 2018 and was elected in 2019 to the National Academy of Medicine of the United States for her outstanding achievements in the fields of medicine. Furthermore, Dr. Iwasaki is well known as an ardent advocate for women and minorities in the sciences. She has spent the past few months trying to understand the immune response in COVID patients. It is a privilege and an honor for us to have the opportunity to listen to Dr. Iwasaki today. So thank you so much, Dr. Akiko Iwasaki, for having accepted the Argentinian Society of Immunology invitation and to share your data with us.
Thank you so much, Dr. Turbani, for that very kind introduction. I'm going to share my screen now. Okay. You can see this? Yes?
Yes.
Okay, thank you. So it's my pleasure to be here with you today for the Society of Immunology for Argentina. I'm going to talk about immune response to SARS-CoV-2 and give you some of the latest updates on what we are doing. And so before I do that, I wanted to give you just a brief introduction to the virus and some of the key issues that surround this virus infection. So as you all know, the causative agent for COVID-19 is SARS-CoV-2. It's a coronavirus family member, and it has a very large genome size for RNA, positive strand RNA of 30 kilobases, and it's also enveloped. And a lot of the discussion around the virus is currently surrounding the spike protein, because this is the protein that is very antigenic and our majority target for antibody responses. But there are many other protein antigens that T cells can recognize. Coronaviruses have caused infections throughout the years, especially these common cold versions of the coronavirus. However, there have been two other very lethal infectious coronaviruses, which are zoonotic coronaviruses, SARS-CoV-1 as well as MERS-CoV, that have infected humans. But obviously this SARS-CoV-2 has really successfully spread throughout the humans, not only because it's a novel virus, but because it has a long incubation period in which people are unaware that they're infected and are being infectious during that period. So the virus actually binds to the human receptor, the ACE2 receptor, with the help of some of the proteases that are co-expressed in the cells. And it injects its RNA into the cytosol, and it replicates, and it also makes viral proteins. And when this successfully occurs in a cell, then it can replicate and make new versions of the coronavirus to infect other cells. So there are multiple places in which we can try to interfere in terms of the infection. And many of the antiviral agents are being currently developed to try to target some of these replication processes. And when you look at the disease-causing pathogens of respiratory virus infections, obviously COVID-19 is the one that I'm going to focus on today, but it really has a pretty, infectious, contagious nature that is more than the flu virus, which has a basic reproductive number of about 1.3, whereas it's estimated between 2 to 2.5 for COVID-19. And case fatality rate really depends on the population as well as the age and demographics, but it's up to 3.4%. And as I mentioned, this incubation time is quite lengthy for this virus. That makes it very difficult for public health measures to be implemented because people who are infected and are infectious are unaware of their status. So, what can the history of virology tell us about how we can deal with this infection? And so we know from the earlier studies with the common cold versions of the coronaviruses that antibodies typically last for about a year. And this has caused a lot of sort of anxiety among the population, people thinking that perhaps there will be no good vaccines that are available to fight the COVID-19. However, people who lost the antibody are still relatively immune to even the common cold coronaviruses. They can get reinfected, but they are protected mostly from the disease. So that's encouraging that even with the low antibody levels, that people are still able to protect themselves with other types of immune responses, such as potentially T-cell immunity. And then the other thing to remember is that, for the SARS-CoV-1 and MERS viruses, there are variability between people with respect to the longevity of antibodies. But there have been antibody maintained for, 10 years or so in some of these people. It's possible to do. And vaccines against coronaviruses have not been made, have not been approved yet, but there are many, many candidates, over 150 candidates that are currently being in various stages of trial. And I'm very hopeful that some of these vaccines will provide a much better long-lasting immunity than the natural infection with SARS coronavirus 2. And there's also this potential seasonality associated with COVID-19, but currently we don't know because we've only been through about 6 or 7 months of this infection. However, in general, there is a seasonality to respiratory virus infections, including the influenza virus, the rhinovirus, and the common cold version of the coronavirus. And that has to do with several factors. Some of it being host intrinsic, such as temperature and humidity that affects the ability of the innate immune system to clear the virus. It is one possible mechanism for the seasonality. And then there is the environmental factor, such as the temperature and the humidity within the indoor environment that we spend 90% of our lives in. Which can affect, again, the respiratory tract and the innate immune response to control the virus. And so these are some of the things that we should be thinking about going towards the winter time, that, some of these environmental factors are controlled, can be controlled to benefit the host immune response as well as virus stability, by maintaining temperature and humidity in the optimal rate. So these are some of the things that we've known before we even got this COVID-19 pandemic. However, there's a lot that we don't understand about this virus. And so what kind of research needs to be done? Well, there's so many. I've listed only a few of them here. But, the immune response to COVID-19, what confers protective versus pathologic response, is really a key issue that we are actively engaging in trying to understand. And that will be the discussion that I will have today with you. Development of antiviral agents would be key in preparing for future pandemics, as well as dealing with the current pandemic. Development of safe and effective monoclonal antibodies is another key way to fight this pandemic, which I know that the first phases of clinical trials are being conducted with some of these monoclonal antibodies. And obviously drugs and biologic to promote disease tolerance is another way to, deal with this infection. And I'll show you today that some of the severe diseases may be caused by the immune response rather than the virus infection itself. And then obviously development of safe and effective vaccine will be absolutely key for us to return somewhat to how we used to live before this pandemic. So just briefly, my lab has been really working around the clock to try to deal with this COVID-19, trying to understand the immune response to this virus. And some of them are listed here. And so today I'm going to talk mostly about human immune response in patients that have enrolled in our study. And we've also created a mouse model of COVID-19, which I'm going to touch on as well, because these two things are complementary. And then we also have other studies looking at the extra respiratory tract infections and potential infection and a pathology that might occur with this SARS-CoV-2, including neuroinvasion and neuroinflammatory consequences of this infection, as well as understanding impact of this infection during pregnancy. But I'm mostly going to focus on the first two topics today. So we've generated a mouse model of COVID-19, and this is in trying to understand both pathology as well as immune protection against this virus. And even though human studies are absolutely key in understanding what's going on during the infection, animal model provides us with a tool to really dissect these at the molecular level. So, this just came out last week in Journal of Experimental Medicine. And what we describe in this paper is a very easy and versatile model of infection where we transduce animals of any genetic background of choice with human ACE2 that is encoded by AAV. And then we wait 2 weeks for the expression of AAV to occur in the lung. And then we infect these animals with SARS-CoV. And then we can measure viral titers and immune responses in these animals over time. And what I'm showing you here is the viral genome load in the infected animal, either that is controlled animal or AAV transduced human ACE2 transduced animals. You can see orders of magnitude higher levels of the virus replicating with the human ACE2 expression. And also this is looking at the infectious viral titer. You can see that only with the human ACE2 transduction can we get infectious virus replicating in the lung of these animals. And we also can use this animal model to monitor adaptive immune response. This is looking at antibody against the spike 1 of the SARS-CoV-2. And it's pretty remarkably quick in terms of the time course of this antibody development in these animals. Within 7 days of infection, we get a really robust antibody response. So we can potentially study the role of adaptive immunity and vaccine strategies using this animal model. And if you look at the expression, infection of the lung in these animals, so the top panel here is looking at 2 days post-infection. The bottom is 4 days post-infection. And just looking at the N protein of the SARS-CoV-2 staining, you can see that the infection is occurring. You can see these red signals indicating infected cells in the lung. And you can also see that this is also spreading around 4 days post-infection. And they seem to be occurring in the alveolar epithelial cells, but perhaps other types of cells are also infected. And EpCAM is just indicating the epithelial location here. So you can see these large bronchus stained here, and then the alveoli stained in dim expression there. So this is sort of what happens during the first 2 to 4 days of infection in these lung. And then when you look at the pathology that occurs in these animals, and this is looking at the human ACE2 transduced versus control animals, and you see that within as early as day 2 post-infection, you can see a lot of these immune cell infiltrating the lung, and that continues for day 4 post-infection. And so this allows us to look at immune infiltrates, chemokines, and the role of different cytokines in infection and disease. So the paper really focused on the role of interferons, type 1 interferons, in resistance versus pathology after this infection. And so this is a review that a student of mine and I put together about what's known about countermeasures by coronaviruses. And basically, as with any successful human viruses, the coronavirus encodes multiple genes to interfere with signaling and recognition, as well as interferon production, as well as interferon receptor signaling, all the way along the path of innate induction of interferon and interferon signaling pathways. So, it's been known from looking at SARS-CoV and MERS-CoV that these beta coronaviruses do encode for multiple evasion molecules. And so we wanted to understand what the consequences of interfering with host innate immune responses by blocking or knocking out interferon IRF3 and 7, which are integral transcription factors that drive the type 1 and type 3 interferons, as well as the type 1 interferon receptor, to see what would be the consequence of virus infection. And if these signaling pathways are key in containing the virus, you would expect the knockout mice to have a lot more virus replication and pathology. So first we examined what the genes that are upregulated during infection in the animals that were first transduced with human ACE2. And many of the genes that came up, you can see, are familiar interferon stimulated genes. And there are, top upregulated genes are in this category of genes. And there are many interferon stimulated genes that are known to be inducible through type 1 interferon. And these overlap with some of these other interferon groups. However, vast majority are contained within this type 1 interferon inducible ISGs. And here are, human RNA-seq experiment in which others have reported the expression of these ISGs, and many of these are overlapping with the mouse model that we have created. And then, so going back to the question of what happens when you knock out key innate transcription factors, IRF3 and 7, or the interferon receptor itself, what we saw was that there was very little difference in the plaque assay here, the infectious virus that's generated in these knockout animals compared to the wild type animals. And this led us to the understanding that virus is likely interfering already so efficiently with the process of interferon induction, as well as its inhibition, that further knocking out the host genes that are responsible for these don't really make much of a difference. But what was kind of striking to us is that when you knock out these key transcription factors for interferon or the interferon receptor itself, there was a significant drop in the recruitment of inflammatory leukocytes into the lung, including this inflammatory monocytes and monocyte-derived macrophages, as well as T cells and NK cells. Their recruitment was quite blunted by the absence of the interferon receptor and to some extent the absence of IRF3 and 7. Whereas neutrophils, there was no significant difference between these genotypes. So what these set of animal experiment has told us is that, first of all, we developed this versatile AAV transduction system to be able to study COVID-19 in the mouse model. And then the type 1 interferon and interferon stimulated genes are induced during the infection. However, when you knock out these key receptor or the transcription factors that are responsible for interferon, we basically see very little difference in viral replication, and yet there's a huge difference in the recruitment of leukocytes. According to the previous results of SARS-CoV-1 and MERS, there was this phenomenon that was observed that rapid induction of interferon corresponded with viral control and mild disease, whereas delayed onset of interferon response, as well as perhaps lower levels of interferon in general, contributes to uncontrolled viral load and viral persistence, and the fact that interferon itself was actually becoming a pathologic, leading to severe disease. So our animal model indicates that perhaps this is the scenario that's happening in the COVID-19 infected host. So armed with this insight, we also looked at human immune response to SARS-CoV-2. And before I get into the data, I want to describe why we were enabled to do the experiments, how we were enabled. So in early March, several of us got together, including Dr. Albert Koh from the School of Public Health, to create a biorepository for COVID-19 patients. And this was an absolutely key organization that allowed us to start collecting samples from patients that are coming into our Yale New Haven Hospital. So every 3 or 4 days, we can collect blood, saliva, nasopharyngeal and oropharyngeal swabs, feces, urine, and everything we can think of that would inform us about what's going on in these patients. And so we had 3 different cohorts of patients that enrolled. First is the inpatient cohort. Obviously, these were sick patients that were admitted to the hospital. And then the second cohort of patient, which we are calling outpatient cohort, who have milder diseases and are not hospitalized, but yet they are COVID positive. And the third cohort is the healthcare workers that we were offering to test their, NP swab and saliva for COVID-19 using PCR. And this allowed us to inform the healthcare workers if they've been exposed to the virus and they're infected with the virus very quickly. That allowed people to quarantine themselves if they're becoming infected, because as I mentioned, there is an incubation period And so the symptom alone would take a long time to develop, whereas enrolling these healthcare workers into our study allowed us to kind of quickly detect COVID positive healthcare workers. And secondly, they provide a very important healthy control group. Those that are not infected would also be a great comparison to the inpatient and the outpatient cohort. So what we did is we collected PBMCs from patients that are enrolled in the study. And we've been doing in real time flow cytometry analysis of various leukocytes from the PBMC, as well as looking at their cytokine secretion from the plasma, as well as the viral load in the nasopharyngeal and saliva, and antibody from the serum. And so we've done this longitudinal COVID-19 immune analysis. And as I mentioned in the previous slide, we had this uninfected healthcare worker as a control for the patients that were studied in our study. We were able to recruit 80 patients who had moderate disease and 33 patients who had severe disease, and some of them unfortunately died. So these are the clinical scores that we've devised, which really depended on the amount of supplemental oxygen that were required in the hospital. So clinical score 1 patients did not require any supplemental oxygen, but they were still admitted to the hospital. And the second grade clinical score is assigned to people who needed 1 to 3 liters per minute of low oxygen supplement, and then moderate, and then in the ICU, as well as the mechanical ventilator. So this is sort of the way in which we were able to monitor the clinical score of the patients that were enrolled. So the first thing we noticed from the flow cytometry analysis is, as reported by others, there is a drop in the number of T cells as they become ill. And this is the healthcare workers in green, and then the moderate and the severe patients. Moderate patients are in blue, and severe patients are in pink. And you can see that the T cell numbers drop quite dramatically from the healthy control to the infected patients. And inversely, we see an increase in the monocyte population, especially the inflammatory monocytes. And what we also see is an increase in granulocytes like neutrophils and eosinophils, which is kind of strange. As you know, eosinophils are, really dedicated to fighting helminth infection, and yet we are starting to see elevated eosinophil numbers in some of these COVID patients. And also, when you measure the serum cytokine, you see an elevation in IL-1, IL-6, interferons, and many of these chemokines, as well as interferon gamma and IL-17. And this is also important to point out that the IL-18 and IL-1 beta are also elevated, especially in the severe patients, indicating that there is inflammasome activation occurring in these patients. The first thing we learned from this analysis is that the viral load in the severe patients appeared not to be declining during the course of investigation. So this on the x-axis is days from symptom onset, and we're following these patients along this symptom onset. Whereas in the moderate disease patients, they start off with higher levels of the virus, but then eventually they can clear the virus. Most of the patients clear the virus, and some remaining had lower levels of viral load, indicating that perhaps what distinguishes the severe versus moderate cases may be the ability of the moderate case to control virus replication eventually in the nasal pharyngeal analysis. And what's striking to us is if you look at the viral load on the x-axis and looking at various different cytokine levels in the y-axis, we see a correlation between the viral load and type 1 interferon alpha, as well as type 2 interferon gamma, and TNF and trail, indicating that it's the viral load that's driving the secretion of these cytokines. And higher levels of interferon alpha appears not to be able to control viral load in these patients. So this is reminiscent of what we saw in the mouse model, that interferon levels or interferon receptor deficiency really didn't make much of a difference in terms of the viral load. And it's really the virus that's driving the interferon, not the other way around. And interferons and cytokines are elevated in patients that developed severe disease. In this case, we're looking at mortality. So when you take the first 12 days since symptom onset and measure their cytokine levels in the plasma and ask what happened to the level, what is the level of these cytokines in patients who ended up being discharged home versus those that deceased. And what you see again is this sort of elevated interferon alpha level found in people who ended up dying of this disease compared to those that were eventually discharged. And similarly, the IL-1 receptor antagonist level is higher in the first 12 days from disease onset in deceased patients compared to the discharged patients. And you can see a similar trend for other cytokines here. So this indicated to us that there are some cytokines that we can monitor during the first 12 days of symptom onset to predict what might happen to these patients. And very similar trend was found if you look at the correlation between the length of hospital stay and disease severity. So these cytokines are indicative, like a biomarker for worse disease prognosis. And if you look over time of different cell subsets as well as cytokines, what we see here is that, again, the severe cases have continued elevation of various different cell subsets, including monocytes, eosinophils, and neutrophils, as well as cytokines that drive type 1 immunity, such as IL-12, interferon gamma. And also, strangely, there are these sort of type 2 immune responses that are also being maintained and elevated in the severe cases during the course of study. For instance, IL-13, IL-5, and IL-4, and even IgE. And similarly, these types of antifungal immune responses, IL-17A and IL-22, are becoming elevated during the course of infection in the severe cases, whereas in the moderate cases, they are able to either maintain low levels or declining levels of these cytokines. So this appears to be a kind of a disorientation of the immune response against this virus that's eliciting sort of these type 2 and type 3 immune responses. So we wanted to understand what is going on in these patients at large. And so what we did was to allow an unsupervised clustering to reveal distinct clusters of patients that are based on different cytokines and chemokines and growth factors. So what we see here is that this unsupervised clustering generated 3 different clusters of patients, cluster 1, 2, and 3. And that is based on these 4 different signatures of cytokines and growth factors. And if you look at the cluster 1, they are most enriched in this signature A, which has to do with tissue repair growth factors, such as PDGF, VEGF, IL-7, and so on. And cluster 2 patients are enriched in various chemokines and mixed cytokines, including IL-4, interferon gamma, TNF, IL-13. Whereas the cluster 3 patients have the highest level of mixed cytokines as well as type 2 and 3 cytokines, including IL-17, TPO, IL-23, and many different chemokines as well. So what are these clusters mean? And if you look at the age distribution and length of hospital stay, there is no significant difference between the age group, but there tends to be a slightly elevated median age in the cluster 2 and 3 compared to cluster 1. And similarly, there seems to be a little upward trend in the length of hospital stay in cluster 2 and 3. And this was the most striking thing, that if you look at what happens to the patients in these clusters, the cluster 1 patients are able to control the clinical score pretty well and eventually become discharged. Whereas cluster 2 and 3 patients, they develop rapidly worse disease, and some of them unfortunately ended up dying of this infection, as well as a more increased coagulopathy in the second and third clusters, indicating that the enrichment we saw in the tissue growth factor, such as the repair factors, are associated with much better prognosis of disease trajectory, whereas the second and third clusters that had the most elevated chemokines and cytokines of all groups did worse from this infection. And if you look overall at various different flow cytometry and cytokine analyses and ask what are the biomarkers for mortality, so the less here indicates better prognosis, and the worse, the closer to the right of the graph, the worse mortality. And what we saw is that, IL-18 and interferon and IL-10, these are sort of associated most strongly with mortality from infection. So as I mentioned earlier, IL-18, IL-1, these are result from inflammasome activation. And that is resulting in sort of worst biomarker for mortality from COVID-19. So to conclude this section, what we found is that it seems to be a persistent viral load in the severe patients that it's just not going away during the course of infection. And this viral load is driving the inflammatory cytokine as well as interferon responses. And this increased interferon and cytokines during the first 12 days of symptom onset is higher in those patients that did worse from this disease. And there are 4 signatures of immune genes or proteins that can distinguish patients. And the ones that do well from this disease have enrichment in the growth factor that might contribute to tissue repair, whereas the second and third clusters that had worse disease outcome have an elevated mixture of cytokines belonging to type 1 and 2 and 3. So again, I put this slide up, schematic, because it seems to suggest that delayed and prolonged secretion of interferon and perhaps other cytokine is leading to the severe disease. And of course, our study is failing to capture what happens within the first few days of infection because people are asymptomatic at that time and they're not coming into the hospital. But if you just look at what happens later on during disease, this sort of scenario may be what's going on in the COVID-19 disease. So finally, I want to end with a sort of looking at some host differences in what the immune responses, particularly paying attention to sex differences in immune response during COVID-19. And so, as you know, there's been many reports now coming from many different countries that male sex is a risk factor for severe COVID-19. So for example, in the USA, men account for 54% of the COVID-related deaths. In China, also, there were more men who died of infection than women, as well as hospitalization from both China and Italy are, increased percentage in men. So could immune differences be contributing to these risk factor for men? And this is not the first disease, first infectious disease in which sex differences are seen for severity and prevalence. So this is a nice review by Sabra Klein's group that demonstrates that in certain diseases, there is a higher severity in women, for instance, with the HIV influenza, compared to, let's say, MERS-CoV that was more severe in men, and hepatitis B, and bacterial, parasitic, and fungal infection. So this is one of those infectious diseases in which there is more severe disease in men. And so we wanted to study what's going on between these men and women. So the study design, which is really kind of developed by Takehiro Takahashi in my lab, is to first look at the baseline analysis, which is to look at the data from the first collection from patients who are enrolled in our study, who are not severely ill and who have not been given any immunosuppressives. So, for instance, tocilizumab or high-dose corticosteroids, people who are on these medicines were excluded from the baseline analysis simply for the reasons that these treatment alone will affect the immune response, immune measurements that we want to study. And then so this cohort A, that is the baseline cohort, consisted of 39 patients, 17 men and 22 women. And we follow these patients over time. We also recruited healthy donors, the healthcare workers, as I mentioned earlier. And then in cohort B, we extended these analyses to 98 patients at all stages of disease to see how well the correlation from the baseline holds with respect to the sex difference in immune response. So first, we wanted to know whether the viral load or the antibody levels might be significantly different between men and women. And here, we did not see a statistical difference between the men who are orange here and women who are purple. There's the nasopharyngeal swab and the saliva. And you can see that there's no significant difference between the sexes, even though the median seems to be higher in male versus female. And similarly, we did not find a statistically significant difference between the antibody level, either looking at anti-spike IgG or IgM. Again, both male and female patients developed some level of these antibodies, and yet there was no significant difference between these. By the way, this is looking at the first time point of collection, but all of these patients are obviously COVID positive. And the fact that you have undetectable level is because these patients may already have undetectable level because they were in the hospital for 4 days before that. So chemokines and cytokines obviously become elevated during this infection. And there were some chemokines that were highly elevated in both sexes, such as CCL5 and CXCL10. There's a huge induction of these kind of chemokines in the infected patients, regardless of the sex. But there were some factors that were more elevated in male than female patients. For instance, IL-8, which is a chemokine that's a potent recruiter of neutrophils, were elevated in male patients and not in the female patients. And here, IL-18, again, the inflammasome cytokine, was elevated in male patients and not in the female patients. So you see that there's a significant elevation of these chemokine and cytokine in male patients over female. And we also detected increase in the non-classical or the inflammatory monocyte in the male compared to the healthcare worker, whereas there was no difference between the female patients. And if you plot the CCL5 level on the x-axis to the non-classical monocyte percentage on the y-axis, there was a very interesting positive correlation between the CCL5 level and the non-classical monocytes in male, but not in female. So there's only a positive correlation in the male patients of the elevated CCL5 level to the non-classical monocytes. And conversely, when we look in the female patients, we found elevated activated CD4 T cells, CD8 T cells, and more terminally differentiated CD8 T cell in female over male patients. And so this is interesting because now we're seeing kind of elevated pro-inflammatory cytokines, innate cytokines in male, and in contrast, female have elevated T cell activation. And so we wanted to ask whether these first sampling tells us about the future disease trajectory. And so we've divided this sort of cluster into patients that develop worse disease, which are the filled circles, versus those that had stable disease over the course of a study. And again, we are showing you male patients in orange and female patients in purple. And what came out of this analysis is that the male patients that deteriorated in their disease were of higher age group compared to female patients in which there was no difference. And there was also elevated BMI in only the male patients that deteriorated and not in the female patients. And male patients with very low T cell activation did worse with the disease. So I told you that male patients in general have lower level of T cell activation. But if you look at the ones that did really poorly, they had very little T cell activation here, and here. Whereas in female, they had, in general, more activated T cell, and there was no difference in the T cell activation level between the stabilized and deteriorated disease groups. And we also saw that conversely to the male patients, female patients that have elevated levels of chemokines and cytokines, such as TRAIL, CCL5, and IL-15, had worse disease outcome than those that had lower levels. So this is a kind of an inverse correlation that in the case of female, it's the higher level of the pro-inflammatory cytokine that dictates worse disease outcome. Whereas in the male, it's the lower level of T cell activation that correlates with deterioration of the disease. And this is one of my favorite graphs because it tells you a lot about what's happening. If you look at the age versus the activation of T cells, and female patients are again in purple and male patients in orange, what we find is that males who age have lower levels of interferon gamma as well as activation of T cells in general, whereas females, even as they age, they are competent to secrete interferon gamma or activate their T cells, meaning that the women tend to age better with T cell immunity than men. And this could explain, for instance, the correlation of advanced age and disease deterioration in men, but not women. So to conclude this last part, what I've shown you is that men develop more inflammatory cytokine responses than women at baseline, whereas women develop better T cell activation. And men who developed very little level of T cell activation had worse disease outcome. And conversely, women who developed more pro-inflammatory cytokines and chemokines did worse in their disease. And this part of this can be explained by the fact that women age better with respect to T cell activation, whereas men decline in their ability to stimulate the T cells as they age. So I'd like to end here by thanking the entire member of my laboratory who've been working really hard, ever since the pandemic hit us, on looking at not only the COVID-19 disease and immunity, but also helping to implement testing of the healthcare workers and the patients. And that had benefited sort of our community at large. And also, I want to really thank the impact team. This is the team that put together the biorepository from the beginning, starting with Albert Coe, but many others, Saad Omer, Charles de la Cruz, Shelley Farhadian, are clinicians that really are at the forefront of treating these patients, as well as Nathan Grubel, Craig Weiland, and Aaron Ring, who contributed greatly to viral genome sequencing, as well as development of animal models and antibody analyses. And I want to thank the founders who allowed us to carry out the research. And I want to end by really thanking my amazing lab members. I'm so privileged to be working with these, spectacular students and postdocs who are really dedicated to trying to understand immunity to COVID-19 and to help the society by doing this PCR analysis every day, all day long. And so I really want to thank them for their amazing effort and dedication. And I will end here to take questions.
Thank you so much, Akiko. A wonderful talk, really. There are a lot of people following you in YouTube, in our YouTube channel. So thank you so much. And we have many questions. Caro, would you like to start? Do you have something?
Yes. So, Akiko, do you think that there is any relationship in testosterone levels and the outcome of the SARS-CoV dose? Or, for example, in hormone replacement or in young women that the hormone levels can help to control the disease?
Yeah, so there has been a lot of interest in that area. And I know that there are already trials of giving people estrogen, like men estrogen, as a potential kind of therapeutics or even preventative measure for severe COVID. And so we are measuring these hormone levels in these patients now, as well as many other metabolites that might be different between men and women. And again, like, I just want to remind that a lot of the severe diseases are occurring in postmenopausal stages. And so, whether they're all receiving hormone replacement therapy or not, we don't know. But I think hormone is maybe one of the answers, but there are other metabolites that are very interestingly different between male and female.
One, Mariana?
Yes, if you want, that's okay. There are many questions here that the public has been writing in the YouTube channel. Here, for example, they are asking you if you can estimate the proportions of the 3 clusters of patients in the entire population.
Yeah. Well, so we're looking at the very severe cases, right? Patients who are admitted to the hospital and they require, clinical care on a constant basis. So if you look at the general population, maybe up to half of the people who are infected are asymptomatic, right? So that's good for those people, but there are many factors that contribute to developing a more severe disease. So what I'm not capturing here are what happens to these asymptomatic people. How are they able to control the virus without any symptom? What I'm also not capturing here are the patients, outpatients who are never admitted to the hospital, but have nevertheless very bad disease. And there are so-called long haulers who are suffering for weeks and months from this infection with very little we can do at this point. And that's, I think, again, not captured in this study. And we'd like to understand the spectrum of disease from asymptomatic all the way to very lethal disease. And we're only at this end of the spectrum when we're not here and we're not in the middle. And that's sort of our next goal, is to understand because the vast majority of people either asymptomatic or mild or having this terrible long hauler disease. And what distinguishes these people is our next goal.
Mm-hmm.
So other people are asking, they say, dear Dr. Akiko, it is interesting to see the trend in Th2 responses and the patients using the studies. Were patients with health history taken into consideration, such as allergies and other morbidities?
Exactly.
So do you think, for example, that the IgE that you find is specific against the virus, or is just specific against wherever this person is allergic to or wherever?
That's right. So we did look at that, whether IgE is specific to the virus, and it appears not to be. So it's interesting what this IgE is, against, or where, like, why they're being triggered, right? And so far, there is no obvious connection between, like say, pre-existing, allergy conditions or anything like that. So we're again trying to develop a more unbiased analysis to be able to say what is correlating with the type 2 disease, type 3 disease, and the fact that each patient has a mixture of these, right? So they're triggering all of these at once. And so how is a viral infection inducing this? This is a big mystery.
Thank you. Here's another question. They're asking if, do you think that the lymphopenia, especially if there is a Treg lymphopenia, that could contribute with the maladaptive cluster 2 and cluster 3 response?
Yeah, absolutely. So the T cell lymphopenia may be a big key in understanding all these misfiring and maladaptive immune response. And so this declining T cells will affect, obviously, our ability to mount a good, antibody response and cytotoxic T cells, and, everything else is going to be affected, including Tregs, as mentioned. So obviously, if you have very little number of Tregs, you cannot control these all these effector immune responses. So this might be contributing to the misfiring. Absolutely. Yeah.
Another one, this is based on your longitudinal studies, do you think that the interventional therapeutics should be applied to control different cytokines together? Which cytokines do you consider are the best targets? And which would be the window period you consider these strategies should be applied to be effective?
Really good question. So, when I talked about the biomarkers of mortality, what we see is, this IL-18 is like number one, as well as sort of all these other, inflammasome related cytokines, and also type 1 interferon. Now, I would not recommend inhibiting type 1 interferon because, it is also required for viral control, and it's a little dangerous to be interfering with that. But inflammasomes, on the other hand, probably will not compromise the host's ability to sort of control the infection, but it's probably contributing to a lot of the pathology. So inflammasomes are one target that I would, kind of predict to have a good outcome. The other thing that this question asks is the combination of cytokines. So I think absolutely that right now, a lot of the trials are only targeting one cytokine at a time. And if we can target multiple cytokines, especially during early stages, before it gets to this sort of ridiculous cytokine levels, would be beneficial. And that's why the first 12 days of symptom onset, that kind of analysis was informative, because it tells us that, even then, even that time period is enough to predict what would happen. And if we can interfere with these cytokines or inflammasomes or whatever earlier, perhaps it would have a better outcome.
Excellent. And what do you think, what is the real cause of lymphopenia? Because there are so many things that have been written regarding many hypotheses regarding the lymphopenia, and I was wondering what was your idea, what was the actual cause of this pronounced lymphopenia in severe COVID patients? What can you tell us?
That's a great question. I wish I knew the answer. I wish I am really interested in that question, and members of the lab are actually actively looking into what causes lymphopenia. You know, direct infection of T cells was proposed. We don't think that that's what's happening. It's probably something other than, like, you know, direct infection and killing. So that, you know, we're actively looking at this. It's such an important question. I don't know.
Okay, thank you.
Another one says that, thank you, Dr. Iwasaki. Considering that the viral load and persistence seems to drive progress to severity, have you assessed antiviral IgA in mucosa from the upper respiratory tract or in the nasopharyngeal?
Yeah, that's a really good question too. And you know, before the COVID, I was really looking into IgA as a potential for, you know, influenza virus immunity, and it's we're still continuing to look at these issues. I think absolutely, if you have high levels of IgA in the upper respiratory tract, maybe you can prevent the spread of the virus from upper respiratory to the lower respiratory. And, you know, so that's sort of, again, the next stage is to really look at mucosal immunity. And so this study focused on the plasma because it was accessible and easy to measure, but that's definitely where we have to go next, mucosal immunity. Yeah.
I think one last question.
Okay, sure.
I was wondering regarding your mouse model, have you tried any of the vaccines that are going on to see if they protect in your mouse model? Do you have any data or any clues?
Yeah, no, that's a great question. We cannot get a hold of the clinical versions because that would be, I don't know, the companies are not going to release those. But yeah, I mean, in general, right? Like mRNA versus DNA versus protein versus inactivated versus, you know, attenuated. I mean, what are the sort of the range of immunity that they induce and whether they're protective? Yeah, this is very key question as we are going forward with the phase 3 trials now. So yeah, we're starting to do some of these basic analyses in the mouse model. So maybe next time I speak, I can say something. Yeah.
Perfect.
So here, Dr. Akiko, they ask you if in a pregnant woman who acquires SARS-CoV-2, the baby would be tolerant to the virus, or how tolerance would work in this case.
Yeah, well, we don't know the answer to that yet. What we've been studying is sort of the impact of maternal infection on the placenta and the you know, the development of the fetus. And thank goodness, most women who are infected with COVID during pregnancy carry the pregnancy to term and have healthy babies. So far, most women are able to do that, but there are cases in which there are severe infection. And we've reported one that in which the placenta, the syncytial trophoblast layer itself was infected by the virus. And it was so dramatic, the infection, that unfortunately the mother lost the baby. But there are cases in which this can happen. And so again, these are the different spectrum of disease. We're looking at the very severe cases, but what happens to the babies that are born and what kind of immunity they carry. Obviously, if the mother is making antibody and you know, the antibody can transfer to the baby, and they might be resistant in the first few months of their development. But we don't know these yet, how well the antibody can confer a protection in the baby.
And in this mother that you find like high viral loads and she lost the baby, do you think that there is a correlation between, for example, NK regulatory cells that are there or Tregs in the placenta, or there is any correlation?
Yeah, so that's the thing. It's just one case that we reported. There has been another case like this reported elsewhere. So it's a very rare event that could happen. And unfortunately, we don't have enough tissue to figure out, like, you know, what went wrong, especially like before the infection, because, you know, not every woman obviously suffer from the same type of infection. So, you know, the fact that the placenta was infected suggests that there might have been some sort of blood-borne virus in this woman, and how that happened and what dysregulation had occurred to make that happen. Unfortunately, these are like things that happened already, and we can't really tell what's going on.
You're having a lot of positive comments here. Very nice talk, impressive work, excellent talk. So there are a lot of very nice
Thank you so
comments.
much. Thank you.
Okay, thank you so much, Dr. Iwasaki, for this amazing and stimulating presentation. We are grateful for this webinar, and we look forward to hosting you in Argentina after the COVID-19 pandemic can be overcome.
Thank you very much. It's such a pleasure to be here. Thank you for all the questions and enthusiasm. Really appreciate it.
Okay.
Thank you. I would like to thank Carolina Mezcua for the effort to make this webinar possible, and also you, Mariana and Carolina, for chairing this session.
Okay.
Okay.
Thank you. I hope we see you
Thank you.
someday in
Bye,
Argentina.
Akiko.
Thank you.
Bye-bye.
Take care. Bye.
Bye.
Thank you.
Bye.
How am I?